Hello guys, so we are going to go over the putting it together, which was your assignment from yesterday, just to make sure that you guys understand why meiosis is important, what the purpose of it is, and if I recommend rewatching those Amoeba Sisters and the modeling videos that we did for your project that you'll be assigned today also. Doesn't hurt to rewatch some of those videos to help you out. Okay, so first of all, it had you count the number of chromosomes in each sperm and egg cell. And what you'll notice is there were half the number of chromosomes in each sperm and egg cell. So 23, if you went through and counted, there are 23 in sperm cell, 23 in egg cell. And then we doubled that for the baby cell. And what this diagram is showing you is that basically you're combining the sperm and egg cells chromosomes to make the baby cell, which makes sense, right? We know that it takes a mommy and a daddy, etc., etc. And you result in a combination of chromosomes, which makes a baby cell. Remember, we call that diploid. Remember these terms? I mean, it's a review, but these guys are haploid. They have half. Haploid equals half. And then the baby cell would be diploid, which is like the normal amount of chromosomes for all of your body cells. So this is what we saw when we looked at karyotypes. You saw 2 copies of each type of chromosome, and it equals 46 in total. From the crossing over, so moving on to number 3, crossing over, and I try to make a drawing for you guys to kind of help you out a little bit. So let's focus on this side. So we have 2 copies of each chromosome for mom and dad, right? So I drew a little M here for mom's copy and one for dad's copy. These are replicated chromosomes. Remember, we had to replicate them before we could do meiosis, just like mitosis, same thing. You have to replicate the DNA, and that's why they're in X form. They are called sister chromatids when they're replicated. And these 2 sister chromatids, so this little finger guy right here and this other finger guy from mom. So these 2, when you put them together, that makes a replicated pair. So think of them as separate chromosomes, separate chromatids apart. But they are identical if they're like in an X shape. If they're in an X shape together, that means they are identical. Each of the little fingers are identical. It means they're replicated. So mom's copy here, both of mom's copies are identical to each other, and both of your dad's copies are identical to each other. So we call them sister chromatids when they're identical. They're called a homologous pair together. That's what the HP stands for, is that homologous pair, because they're the same type of chromosome, which is why you have, remember, 2 copies of each one. They're the same type of chromosome, but they each have a little bit of different information. You want your mom and dad's chromosomes to have different information, right? I mean, that makes sense, unless they're very closely related, which you don't want to happen. They should have very different DNA from each other. So mom and dad's copies are different from each other, but the same type. What I mean by that is, if, for example, if this is chromosome number 3, so say it's chromosome number 3 over here, it's this type of chromosome. The eye color gene will be found on both mom and dad's copies, but the eye color type they have might be different. So mom might have the gene for brown eyes, where dad might have the gene for blue eyes. But for these, for the homologous pair, both the gene for eye color is on these chromosomes. So we call it a homologous pair when they're the same type of chromosome, but you have one from mom, one from dad. So they're going to have a little bit of a different information on there. When they do crossing over, just the ones that are right next to each other are going to switch. So remember, we did the pop beads. We took one off and added it to the other one. And then at the end, that's what this is showing. Basically, you're switching the ends of just one of those sister chromatid pairs from each pair. And then you get this to where you have 4 little finger guys, 2 X's. Remember, 2 fingers make an X. You have 4 finger guys, and each of them has a different piece of information. Okay, so this one, this little finger, different from this finger now because she has this chromosome. Mom's copy has a little bit of dad's copy on there. This dad's copy now has a little bit of mom's information on there. And then this last finger is just all dad's. So each of those fingers, each of those chromatids, 4 chromatids, 1, 2, 3, 4, is a little bit different now, which matters. Remember, if you go back and look at the modeling meiosis video, it matters because when you're separating them into 4 different cells at the very end, it means that you're going to have each of those cells be a little bit different. So crossing over is one way that we get variation in meiosis. So how does it make them different? What is crossing over? It's when you switch these little finger pieces. So that's homologous pairs. When you switch a little bit of the tips of one of the sister chromatid pairs. And this creates variation so that each chromatid is different from the others. Okay? Creates a little bit of variation. All right. So now we move to the second way we get variation in the cell in meiosis, and that's independent assortment. And this goes through really well what independent assortment is. So basically what it's asking you to do is, how's the top image in this picture, how's the top different from the bottom? And if you look at these very closely, the top has these guys switch. I circled it for you in pink so you could see. So our Y's are switched. Here it has the big Y's on top and then little Y's on bottom. Remember, they're sister chromatids. Like both of these are going to be the same, both of these fingers, because they're replicated. They're identical sister chromatids. They're exactly the same. And this is its homologous pair. Basically, these guys are switched. Whichever one's on top, which one's on bottom. So here we have 2 big Y's on top. Here we have 2 little Y's on bottom. And it's switched here, 2 little Y's on top and 2 big Y's on bottom. And just by switching the order in which they're arranged, you can get different outcomes. So if you look all the way, oops, it's like looking in a mirror. It's kind of hard to do. If you look here, try to put that a little closer to you. Each of these is a little bit different. So compare here to here, you get different results based on the order they're in. And they're basically shuffled. Each time you go through meiosis, whether you're making a sperm or egg cell, those chromosomes are shuffled, so you never know which order they're going to be in. And remember, we have 23 of these different versions. We're going to have 23 of these going on. So you're going to have a lot of different ways you can get variety. Okay? Also, if you're confused by the orientation of this, we did it like this yesterday, and what you'll see in most diagrams is you'll see it like that. So don't be confused by the way they drew it. I thought that was kind of a little wonky myself because most pictures are that way. But this is how we drew it yesterday. So basically now you're just switching the order in which you put these. Which one was on the right versus which one was on the left. And like I was saying, they are shuffled of which one is on the right, which one's on the left. So at the very end, you do result in different things based on the order you had them in. And it says, have it upside down. So you have, for number 5, you have chromosomes on the right or top. If you're looking at it this way, it'd be chromosomes on the top are going to be switched. And which one's on the right, which one's on the left, which results in different combinations of chromosomes in each of the cells. For number 6, it says, could you arrange this any other way? Well, yes, you could switch the G's. So you could switch these guys, you could put in a different order. But if you think about it and if you draw it out all the way, it's not going to result in anything different. So really, these are the only 2 ways if you're only using 2 chromosomes. These are the only ways you can get different orders with 2 chromosome types. But say we add 23, like real human cells have 23 chromosome types. So what if we had a big diagram like this? We did this with all 23 types of chromosomes, which would be a very big picture. And it's usually we only use 2 because it simplifies it a little bit. But if we use 23, then there's a lot of different orders that you could have of each of these. So a lot of different ways you could arrange them. In fact, the way you can calculate that, you basically have a 2 to the 23rd power is how many combinations you could get out of that, which, if you're wondering, is about 8,388,608. So that's how many different combinations you could get just from the rearranging of the order of the chromosomes, which copy is on the right versus left. So with 2, you can't really get any more different combinations, but with 23, a lot more combinations can be made. So we have then that leads to number 7. So why is it beneficial for these sex cells to be made to be different from each other? Why is it important that each of the sperm cells are different, each of the egg cells are different? Well, we create variation in our human species and population based on this way, right? If you didn't have variation, all of your siblings would be identical clones to your parents, which would be weird, first of all. But you'd have it from a survival standpoint, it makes sense. Remember, the evolution acts on variation. So the only way we can adapt to a changing environment is if we have some that are more adapted to the environment than others. You have to have variety. For example, a pandemic, timely, right? In a pandemic, you have some people who are going to survive because they have a great immune system for attacking that virus. Whereas some are going to have a weaker immune system for that. And that's a genetic issue. It's environmental to some extent, but the way your immune system reacts is genetic. So if you have some that have great genes for fighting off a viral infection, whereas others don't have great genes for that, the ones who don't have great genes are going to be susceptible and die, whereas the ones with great genes for fighting infection are going to survive the pandemic. However, what if we were all the same and we all had weak immune systems for a particular virus or bacteria or whatever? If that were the case, then it would wipe out the entire human species. Well, that wouldn't be very good for us as a species, and it would mean that no species would be able to survive or adapt to their environment, which is why variation is important. Okay? And why is meiosis necessary then? Because via crossing over and independent assortment, both those two things, you create variation. Okay? So that's it for today. there's gonna be 3 videos today. Make sure you watch all 3 because each of them are important. Okay. Bye.